So welcome to Poster Spotlight Session 3. This is Highlights on Novel Therapeutics. My name is Sara Tolaney. I'm a breast medical oncologist at Dana-Farber Cancer Institute. So this morning we will have two different poster discussion sections. So the first discussant will go, and then we will have a question and answer session where the audience can ask any questions of our poster presenters and discussant, and then we'll go to our second poster discussion and then follow again by Q&A. So I'd like to welcome our first discussant this morning, Dr. Heather McArthur, who runs a breast program at UT Southwestern. And today she will be discussing innovative immunotherapy approaches in breast cancer. So thank you, Heather.
Great, thank you, Sarah. So these are my disclosures. So I had the pleasure of reviewing the first four posters, and I just would like to set the stage. I don't really need to tell this audience, but just as a reminder, the Keynote 355 study was the randomized phase 3 study that randomized patients with metastatic triple-negative breast cancer in the first-line setting to receive chemotherapy physician's choice with or without pembrolizumab, and became a standard of care based on this overall survival advantage, 7-month delta in favor of pembrolizumab. It's worth reminding ourselves, though, that this benefit was seen only in patients who had PD-L1 positive tumors as defined by a combined positive score greater than or equal to 10, and less than 40% of patients had PD-L1 positive tumors. So this is an opportunity that's available to the minority of patients, not to the majority of patients. And one of our other learnings from the application of immune therapy is that the earlier on we apply these strategies, the more likely they are to succeed with much more modest responses in the second-line setting and beyond. So first-line approaches are critically important, but there is still certainly an unmet need for metastatic triple-negative breast cancer. How do we make these strategies relevant for the majority instead of the minority? So in the first poster, tislelizumab plus sitravatinib and paclitaxel are explored in patients with untreated, locally recurrent or metastatic triple-negative breast cancer. And by way of background, tislelizumab is a humanized monoclonal antibody against PD-1. Sitravatinib is a somewhat complicated molecule. It's an oral spectrum selective tyrosine kinase inhibitor that targets the TAM and split family receptor kinases plus MET. And so what does that mean? Well, TAM receptor tyrosine kinase inhibition may promote the depletion of myeloid-derived suppressor cells in the tumor microenvironment and repolarize tumor-associated macrophage toward the pro-inflammatory M1 phenotype. So it changes cells within the microenvironment to a more pro-inflammatory condition. The split receptor tyrosine kinase inhibition may reduce immunosuppressive T regulatory cells and myeloid-derived suppressor cells within the tumor microenvironment. So the net effect of all of those targets is that it may reverse resistance to immune checkpoint inhibitors and augment the anti-tumor immune responses. So this is the SPARK study design that's discussed in this poster. It's a multi-cohort, two-stage design, a phase 2 trial looking to evaluate the efficacy and the safety of this combination of drugs, tislelizumab and sitravatinib, together with nab-paclitaxel in the first-line setting in cohort C. So we're focusing on that cohort from the SPARK study in the red box there. Primary endpoint was overall response rate, or one of the two primary endpoints was overall response rate, and you can see here that the majority of responses are partial responses, but the net overall response rate is 75.7% with a disease control rate of 97.3%. And just as a reminder, the disease control rate, using a slightly definition, and of course cross-study comparisons are to be discouraged, but just again to set the tone, the disease control rate in the Keynote 355 experience was 56%. And the overall response rate in the Begonia study with Dato-DXd and durvalumab was 79%. So just for context. So what is the cost of potential innovation? Well, one of the costs is not just, of course, financial, but of course toxicity. So toxicity or treatment-related adverse events occurred in 97.3% of patients. 43.2% experienced grade 3 or higher treatment-related adverse events. palmar plantar erythrodysesthesion, 8%, elevated ALT in 11%, and 3% of patients had either low white blood counts, low neutrophils, elevated AST, urinary tract infections, or hypertension. And looking at biomarkers that might be predictive for response, patients who had CD8 positive T cells in the microenvironment were more likely to derive benefit. And interestingly, in the Kaplan-Meier on the right there was no impact by combined positive score for PD-L1 using the cutoff of greater than 1. So moving on now to the second poster, this is a study looking at the induction of cisplatin, Abraxane, and pembrolizumab, followed by pembrolizumab with olaparib as maintenance therapy in metastatic triple-negative breast cancer. This is the complement study, and again, Keynote 355 is the standard of care for patients with PD-L1 positive tumors using a combined positive score greater than or equal to 10. The role of maintenance induction after Keynote 355 is not known. However, we do know that olaparib is effective in platinum-sensitive populations, and we know from the Topasio and Mediola studies that immune checkpoint inhibitors together with PARP inhibitors are synergistic and can be co-administered safely in metastatic triple-negative breast cancer. I would just remind you that we have seen a study before Keynote 355 was approved. This was the brainchild of Rebecca Dent, the so-called DORA study. This study enrolled patients who were PD-L1 unselected, who received at least 3 cycles of chemotherapy, platinum-based chemotherapy, and who had at least stable disease or some response, and then randomized them to receive olaparib versus olaparib with another checkpoint inhibitor, durvalumab. And what they observed was excellent durable responses in both the olaparib maintenance-treated population as well as the population treated with olaparib together with durvalumab. So again, this predated the Keynote 355 era. These are patients who were treated with chemotherapy without immune therapy as induction and then went on to olaparib with or without immune therapy. So this is the complement study design. It's an ongoing randomized open label phase 2 study. It is ongoing, so 52 of the 132 planned patients, or 39% of planned patients, have been enrolled, with 29 out of those 52, or 56%, being randomized. Patients are enrolled if they have a PD-L1 combined positive score greater than or equal to 1, so a different threshold for defining PD-L1 positivity compared with Keynote 355. Patients receive induction chemotherapy in the form of cisplatin with nab-paclitaxel and pembrolizumab, so sort of a riff on the Keynote 355 regimen. For patients who have stable disease or a response, they're randomized to receive olaparib with or without pembrolizumab, and the primary endpoint here is progression-free survival. So of the 52 patients enrolled, the majority, almost 80%, had triple-negative breast cancer at their initial early-stage diagnosis, but 21% had hormone receptor positive breast cancer that converted to triple-negative breast cancer at recurrence. Patients were evenly randomized between the 2 arms. Notably, median progression-free survival has not yet been achieved for both groups, which makes the interpretability of the early projections for PFS a little bit challenging. But so far the data looks encouraging, and interesting to note that patients with conversion to triple-negative breast cancer, so those who are hormone receptor positive who converted, seem to derive less benefit from this maintenance strategy than those who were consistently triple-negative, with a doubling of progression-free survival. I'm going a little bit out of order, so this is the 4th poster, so I'm looking at all the metastatic studies, and then we'll end with the curative intent study. So this is a study looking at overall survival with a vaccination strategy, the BRIA IMT allogenic whole cell-based cancer vaccine. This vaccine has 3 components: SVBR1-GM, which is an experimental cell line that's modified to stimulate the body's immune cells to attack breast cancer cells. It incorporates cyclophosphamide, and it incorporates interferon. This is an ongoing prospective phase 1 to 2 study, adding the vaccine to PD-1 blockade. There's 2 different choices there in metastatic breast cancer in all subtypes. And according to a press release from September 2024, the combination elicited an overall survival benefit among heavily pretreated metastatic breast cancer patients. So let's take a look at that data. So 54 patients enrolled, a median 6 prior lines of therapy, so a very heavily pretreated population. 44% having received a prior antibody-drug conjugate, 20% receiving a prior immune checkpoint inhibitor, and 63% receiving a prior CDK4/6 inhibitor. About 2/3 of patients had visceral metastases, and you can see that the clinical benefit rate was observed in all subtypes of breast cancer, and most potentially impressive in the HER2-positive cohort, although notably only 2 patients in that cohort. On the right is a Kaplan-Meier curve looking at introduction of immune checkpoint inhibitor therapy at the onset, so at cycle 1 versus delayed, and you can see at cycle 2, and you can see that there's benefit in favor of early initiation of checkpoint inhibitor therapy. There are different formulations of the vaccine, and the bottom line from the left side is that the formulation without pulsed interferon gamma was superior. So that's the phase 3 formulation, and the side effects were mostly grade 1 and grade 2, which I've highlighted here, and not surprisingly, primarily fatigue, injection site reaction, and some GI toxicity. I also thought this was an interesting data set, a small number of patients with brain metastasis, and you can see the vaccine, either administered as monotherapy or in combination with immune therapy, seemed to have some intracranial activity, which I think is encouraging. So these preliminary results will be confirmed in the ongoing randomized phase 3 pivotal registration trial, and that data is anticipated. And last but not least, we saw a poster or data looking at vaccination with a MUC1 targeting drug that improves survival in patients receiving neoadjuvant chemotherapy. This is results from the SABCS-G34 trial. The synthetic lipopeptide tesomotide is a vaccine designed to induce a cellular immune response to cancer cells that express the glycoprotein MUC1, which is overexpressed in more than 90% of breast cancers. This is a prospective randomized phase 2 multicenter study investigating the efficacy and safety of the vaccination in 400 early-stage breast cancer patients when given concomitantly with neoadjuvant chemotherapy or neoadjuvant endocrine therapy. So 400 patients are randomized to receive the standard of care. So postmenopausal women could receive neoadjuvant letrozole endocrine therapy. Pre- and postmenopausal women could receive neoadjuvant chemotherapy, and they're randomized to receive their standard of care with or without the intervention. The primary endpoint for this study is residual cancer burden after surgery. Follow-up was available for 72% of patients, most of whom received chemotherapy in the neoadjuvant setting. So the group previously reported that there was no difference in RCB 0 or 1 rates with and without the vaccine in endocrine and chemotherapy-treated subgroups. The addition of the vaccine did not affect overall PCR MUC1 expression or TIL content. So again, the RCB was the primary endpoint for this study. Here they report on longer-term follow-up from the study, so secondary and exploratory endpoints. And you can see here that those long-term follow-up data favor standard of care together with the vaccine with an impressive 7 years of follow-up. So you can see an almost 9% improvement in invasive disease-free survival, almost 15% distant relapse-free survival, and impressive overall survival benefit here. So again, secondary endpoints. So although this study was negative for the primary endpoint of RCB, the long-term outcomes, in my mind, justify further exploration of this strategy. So in summary, strategies that target other elements of the tumor microenvironment beyond those mediated by PD-1 or PD-L1 warrant further study. PARP inhibitors with or without immune checkpoint inhibitors may be an effective maintenance strategy for patients treated with a Keynote 355-like regimen or after platinum-based induction therapy, and modern vaccine strategies show promise in both the advanced and early stage settings. Thank you.
Thank you so much, Dr. McArthur. So we'll now open these posters up for questions. So those in the audience in person can come up to the mic, and those who are online, please submit your questions online. While people are coming up, I will just start off with a few questions. So the first question would be for Dr. Singer regarding the MUC1 vaccine. it seemed like it's quite interesting that there's not a difference in RCB, but there does seem to be this long-term benefit in terms of survival outcomes. Were those survival outcomes different by subtype? And then it seemed like maybe high MUC1 may be associated with better long-term outcomes, and is that also different by subtype in terms of expression and outcome?
Well, it is interesting that in the subgroup that received chemotherapy plus minus desemetide, we did not see a difference regarding the tumor biology, and we seem to have the same, we seem to derive the same survival benefit in triple-negative tumors as in hormone receptor positive HER2-negative tumors. In the neoendocrine treated patients, it's a bit different. I mean, there was only around 50 patients who received either desemetide or did not received standard of care alone, which brings us to very small numbers, and even a 7-year follow-up period in a prognostically very favorable subgroup is not enough to show a difference. But in the chemotherapy and in the overall population, we did see this difference, and it was not dependent on tumor biology.
Oh, thank you very much. So maybe, yeah. Oh, I'll take the question at the mic.
Thanks. Ting Bao, Dana-Farber. This is a question for Dr. Sara Chumsri. So interesting results, and what's the overall survival in your study compared to other studies? Thank you.
Yeah, thank you very much. That's a great question. I think that's also the primary endpoint for the ongoing phase 3 trial, which is the overall survival. So in this particular phase 2 trial, the overall survival, the median overall survival is about 13.4 months, which is compared favorably to some of the modern clinical trials. So if you look at the trials like the TROPiC-02 trial, and in this particular trial, the majority of patients are ER positive, HER2 negative. If you compare it to the TROPiC-02 trial, the median survival in that trial was about 14.4 months, and like PFS time on that with the sacituzumab was about 5.5 months. If you do the math, like post-progression after ADC, it was about 8.9 months. And this particular trial, our median regimen was about more than 6, and a majority of patients already had ADC. So with the 8.9 compared to 13.4, it's quite favorable. So hopefully, but the phase 3 trial is ongoing right now, and overall survival is the endpoint. Yeah.
Thank you. The next question is for Dr. Tao regarding the complement trial, which looked at the platinum induction followed by maintenance strategy with PARP inhibition. So one question is, do you have the numbers of patients who have germline BRCA alterations or HRD? And then 2, now we've seen also negative data from the KeyLink trial. Do you think there's any role to PARP maintenance?
Thank you for this question. In our study, the patients' HRD status and BRCA status were detected before the patient enrolled this study. But this is for retrospective study analysis, so it not influence the study ongoing.
Oh, thank you very much. It obviously is very early data, so hard to make any definitive conclusions. So we'll await your longer follow-up with further PFS events. The next question comes for Dr. Tang. You did show activity with this very novel TKI in combination with a checkpoint inhibitor and chemotherapy. What do you think is driving the benefit, given that the TKI has multiple targets? What do you think the rationale is for the activity that you're seeing?
Thank you for your question. I think this combination is very advanced because of the multi-receptor and multi-pathway block. So that may be accompanied with the nab-paclitaxel together to do some benefit to the disease, I think. And the mechanism is still under investigation.
Thank you. Another question for Dr. Chumsri on the BRIA IMT. vaccine. You saw some very interesting data about CNS activity. Why do you think that may be, and what further work is ongoing to understand benefits for response in CNS metastases?
Yeah, thank you, Dr. Tolaney. So I think that is a very interesting observation. Some patients continue to have some depression in the trial, but in the phase 1 and 2 trial, patients with they have to be stable or treated brain mets, so sometimes it's difficult to see whether was it the recent effect of the radiation that the patient have. But on the other hand, it is quite encouraging, and like with the immunotherapy, when you activate the immune system, CD8 T cell can potentially cross into CNS, and there's been some report of other immunotherapy that with the CNS response. So I guess we have to see more data, and the company does have planned clinical trial in the future to look at this specific question, whether patients with perhaps active brain metastases might respond. And there was some patient even with leptomeningeal disease that responded to the treatment. So stay tuned.
Yeah, well, that's very interesting. And one follow-up question, do you think there should be any differential activity based on subtype? Because obviously, you enrolled small numbers of patients in each subtype, so it's hard to differentiate if you're seeing greater activity in one subtype versus the other. And the phase 3 study obviously has all subtypes that is ongoing, but do you think there's better rationale in one versus another?
Yeah, so this particular cell line, SVB01GM, is actually derived from patients with history of HER2-positive breast cancer, so that might be more tumor-associated antigen for HER2-positive. But then the phase 2 trial only had small, 2 patients of HER2, but it is quite promising that both of them responded. And that's the reason why I think in the phase 3 trial, they want to enroll more all subtype of breast cancer. In ER-positive, that's conventionally considered tumor immunologically cold, also seems to have a good response. So I think we will see. And in the phase 3 trial, they're going to stratify according to subtype, and then we'll see which one has more and promising to go forward.
No, very interesting to see activity in such pre-treated patients with the immunologic mechanism. So thank you. Any further questions for these poster presenters? If not, excellent job, everyone. Thank you very much. Well, next we'll move on to our next half of the poster discussion, so thank you all. So if I could call up to the stage the poster presenters for PS05, 06, 07, and 08, and then also welcome our discussant on stage as well. We'll just wait.
Hello, everyone.
Oh, just 1 minute. So if Dr. Wang, Dr. Sung, Dr. Liu, and Dr. Wu could come to the stage. Hi, thank you. Very nice to meet you. Thank you. Thank you. Thank you. Well, thank you very much. So our next discussant is Dr. Thomas Grinda from Gustave, Roussy, who will be discussing abstracts 05, 06, 07, and 08. So thank you very much.
So, hello, everyone. I'm Thomas Grinda from Gustave, Roussy in France. First of all, I would like to thank the organizer, the staff, and the author for their wonderful poster. And I'm very happy to present this discussion, Targeted Powerhouse, the role of bispecific antibodies, TKIs, and ADC in breast cancer. This is my disclosure. So, this discussion will be divided in 2 parts. First, anti-PD-1, PD-L1, anti-VEGF, and chemotherapy combination for patients with metastatic triple-negative breast cancer, with a poster on ivonescimab, PM8002, BNT327, and AK105 plus adnotinib. And then, anti-HER2 ADC in neoadjuvant setting for patients with HR-positive HER2-low breast cancer, with a poster on SHR A1811. So the keynote 355 demonstrates the efficacy of PD-1 blockade with pembrolizumab in combination with chemotherapy in first line for patients with metastatic TNBC with high expression of PD-L1. However, in the intention to treat population, so for all patients, regardless of the PD-L1 expression, pembrolizumab did not show benefit in terms of objective response rate, progression-free survival, and overall survival. So in a pre-Keynote 522 era, so before the widespread prescription of pembrolizumab in adjuvant setting, 60 to 70% of patients are not candidates for combination of anti-PD-1 plus chemotherapy in first line. In the first three posters, the author tested a combination of an anti-PD-1 or PD-L1, an anti-VEGF, and a chemotherapy, because each of these drugs increase the immune response against cancer cells at specific stage at the cancer immune cycle. First, chemotherapy increases immunogenic cell death, thereby stimulates the release of cancer cell antigen. Second, anti-VEGF block neoangiogenesis and promote the tumor infiltration by T cells. And anti-PD-1 reverse the inhibition of immune checkpoint and promote the elimination of cancer cells by T cells. So in this free study, the free study describes a new way of blocking the PD-L1 or PD-L1 and VEGF pathway. Ivonescimab and PM8002 BNT327 are both bispecific antibodies composed by a single chain variable fragment of an anti-PD-1 for ivonescimab and an anti-PD-L1 for the PM8002, and attached to the AV chain of an anti-VEGF antibody. AK105 is an anti-PD-1 monoclonal antibody, and anatinib is a multi-target anti-angiogenic TKI. It's interesting to note that all the three antibodies have no or altered Fc portion because this reduces immune-related side effects by avoiding ADCC and CDC and increase the half-life of the drug by reducing antibody clearance. The three studies are also very similar in design. All phase 2, all in first line for TNBC patients with some study have excluded early relapse patients, all with taxane as chemotherapy, 10 to 20% of patients with a CPS score upper than 10 with a few liver and brain metastases. And to note that only 35% of patients receive taxane in the AK105 study compared with around 60% for the other. And none of these patients receive anti-PD-1 in the neoadjuvant setting. All three treatments achieve a very high response rate, between 75 to 80%, with less than 5% of initial progressive disease and a median of PFS between 10 and 13 months. Most important, for the both specific results were presented according to the CPS score and seems to be independent of PD-L1 expression. So the triplet combination shows significant anti-tumor activity, low primary resistance, and objective response rate and PFS appear to be independent to PD-L1 expression. The toxicities were again quite similar, with mostly hematological toxicity with 20% of grade 3 neutropenia, also hepatotoxicity, including 5 to 10% of grade 3, but also anti-angiogenic toxicity with hypertension and proteinuria. To note, immune-related adverse events were described only for the PM8002 study with 9.5 of grade 3, and one toxic death happened in the AK105 study for hepatitis. So mainly hematological and hepatic toxicity so far. Here is a benchmark of all the phase 2 trial evaluating the triplet of anti-PD-1, anti-VEGF, and chemotherapy in the first line in patients with metastatic TNBC, with a free study presented today, but also with a future C+ study of camrelizumab and fanitinib in a specific population of immunomodulatory TNBC, and also the atractinib study with two monoclonal antibodies, atezolizumab and bevacizumab, in a larger population. Of course, this is indirect and informal comparison, and this must be analyzed with caution, but we can see that the results are consistent with high response rate and a median of PFS around 11 months. It's very promising, even if we remember, but not compare to the result to the phase 3 Keynote 355. So the triplet combination of taxane, anti-PD-1, and anti-VEGF is promising, but in an early, non-randomized, single-country studies. We need a comparative study evaluating overall survival to confirm the triple effect. As remember, in all the phase 3 study in metastatic breast cancer for bevacizumab, but for all anti-VEGF before, they showed a significant benefit in progression-free survival, but at the end, not overall survival benefits. So my open question, we don't know the added value of a bispecific antibody versus the dual inhibition. With the ADCs coming in the first line, which is the ideal partner, chemo or ADC? What will be the anti-tumor activity in more resistant population? So after prior exposition to PD-1 in the early setting, or in early relapse metastatic TNBC patient? And will all patients, we need a triple combination. So we have a crucial need for biomarker for this patient in first line. The last abstract is about SHR A1811 in neoadjuvant setting. SHR A1811 or trastuzumab rezetecan is a novel anti-HER2 ADC composed by a topo-1 inhibitor payload with a cleavable linker and with a bystander effect. Its structure looks like very similar to TDXD. However, it differs in preclinical model with a lower DAR, 6 versus 8, but comparable cytotoxicity, so maybe less off-target toxicity, and a different payload with a higher membrane permeability, so maybe more effective in heterogeneous tumor. What is the rationale to use it in neoadjuvant setting? First, the majority of patients with HR-positive, HER2 non-positive breast cancer have a HER2 low tumor. Second, in patients with HR-positive tumor, PCR rate were slightly lower in patients with HER2 low versus HER2 zero after neoadjuvant chemo. But all patients with PCR low or negative in PCR at a very good prognosis. So the author wanted to evaluate the efficacy and safety of trastuzumab rezetecan as neoadjuvant treatment for HR-positive, HER2 low breast cancer patients. They selected patients with stage 2 to 3A with a Ki-67 upper than 14 and gave 8 cycles at 6.4 milligrams per kilo of trastuzumab rezetecan before surgery. The primary endpoint was the objective response rate. 35 patients in the first stage were enrolled, mainly with score 2+ of HER2 status, mainly in the stage 2, and mainly with a T2 tumor. Objective response rate was high, 74.3%, and validated to go in the second part of the study. However, no PCR was obtained in this study. Toxicity was hematological with almost 50% of grade 3 neutropenia, and also digestive with 11% of grade 3 diarrhea. And no interstitial lung disease was reported so far. As a remember, the trio USB12 talent was the equivalent trial with 6 or 8 cycles of TDXD. Among 22 patients, objective response rate was similar, 68%, but however, there was 1 patient in PCR and 3 patients in RCB 0 or 1, and 1 patient had a ILD grade 2 in this study. So ADC are toxic treatments. The first question is, are all patients with HR positive HER2 low breast cancer are good candidates for neoadjuvant chemo or ADC? Indeed, if we look at the PIM50 subtype among patients with HR positive HER2 low breast cancer, the luminal A subtype was the most frequent. And we know that patients with luminal A tumor have a very low PCR rate, but an excellent prognosis. So we need to look beyond HER2 low status to identify the right patient for ADC in the neoadjuvant setting. In this situation, for example, genomic assay could have been used to select patients for neoadjuvant ADC. The second question is, what benefit and endpoint should we expect from ADC in neoadjuvant setting? In my opinion, there are 2. The first is downstaging to allow less surgery with the endpoint of breast-conserving surgery rate, which has been underused in the recent neoadjuvant trial. The second is to improve survival. And for this, in the NSHAR positive patient, the RCB0 and RCB1 score are validated proxy of survival and should be used as endpoint in neoadjuvant trial. So thank you very much for listening. And I would like to thank all my colleagues, friends, and mentors who helped me to do this presentation. Thank you very much.
Well, thank you for that excellent discussion. So now we will open these abstracts up for questions. So please submit your questions online or feel free to come up to the mic. So maybe one of the first questions I will ask regarding SHR A1811. So Dr. Zhao, it's a really interesting neoadjuvant trial looking at our HER2 ADC in this preoperative setting. And as Dr. Grinda alluded to, the data do look quite similar to Talent, where we're not seeing high PCR rates, but we are seeing radiographic responses. Do you think patient selection is an issue here? You know, as Dr. Grinda brought up, a lot of these tumors could be luminal A. It sounds like you tried to select by slightly elevated Ki-67, I think over 14%, but didn't seem to use genomic assays for selection. Seems like the study is currently ongoing with the same eligibility, but will you retrospectively go back and look at genomic assay scores or other biomarker predictors for potential benefit?
Yes, thank you, Dr. Sara. It's a very good question. Yes, indeed, in China, we didn't routinely use multi-gene tools, testing tools to for the selection of the patient, both for the decision making in clinical practice and in the research. We use the conventional clinical pathological indicators like the histological grading and the Ki-67. Yes, this will impact the result of the research, but we should know that nowadays in many of the clinical trials, the conventional indicators also used a lot. Yeah, thank you.
Thank you very much. So there are a few questions online coming in about the combination of PD-1 VEGF bispecific agents. We saw two very nice posters regarding these data. So maybe I'll ask the first question of Dr. Wang. Regarding the ivonescimab poster. So you do see activity in the PD-L1 negative population. Do you think this activity is being driven by the anti-angiogenic effects of the drug, or what do you think is driving the benefit with these agents in a PD-L1 negative setting?
Thank you. In our trials, the CPS less than 1, about half patient. And most is less than 10. But PFS is equal to about 9.3 months. And also the DOR also is 100% for the no PD patient. So I think it's, this combined, the two targeted combined, is useful for this the TNBCs, and also some patient was the HR convert to the TNBC. So and in the A is very low, no, the IOA and also the EGFR is no. So most is from the chemos. So I say it's the efficacy and safety. Thank you.
Thank you very much. Dr. Wu, you presented some very nice data about BNT327, also a bispecific targeting a checkpoint and VEGF. I guess one question that's being asked from the audience would be, what's the ideal partner to utilize with this bispecific agent? In this case, taxane was utilized, but was brought up by Dr. Grinda. Certainly, antibody-drug conjugates are moving into this space. Do you think an ADC would be a good combination, and is there work ongoing with combinations with antibody-drug conjugates?
Yes, this is a really good suggestion. Maybe in the future we could design some studies for different combination, but till now, the standard combination regimen is chemo agents like paclitaxel or nab-paclitaxel. So I think there might be different mechanisms when you design different combinations, especially when you use the ADC. It would be more sophisticated situation. We need more studies, maybe preclinical studies, to demonstrate the efficacy.
Thank you. I actually have a follow-up question to that with regarding what the optimal partner is, we've seen data suggesting that anthracyclines may be a good partner, certainly as part of Keynote 522 in the preoperative setting. That is a standard component at this point in time of treatment. So the question was, would further work with anthracycline combination be rational?
Well, if we focus on the combination for chemo agents, when you consider the single agent for chemo, I think taxanes would be the best choice, the best accompanied agent when you combine with the bispecific compounds.
Thank you. One question for Dr. Wang regarding ivonescimab was, what was the rate of proteinuria and hypertension with ivonescimab therapy? Yes, the rate of proteinuria and hypertension with ivonescimab.
No patient who has hypertension and urine proteins and also the proteins in this group.
Sorry, there was no proteinuria at all?
Okay.
No protein in the urine?
No. Thank you.
Actually, in our cohort, the proteinuria and the hypertension, most of the patients are grade 1 or 2. Only 5 patients had grade 3 or higher AE like this. So it's manageable AEs in this study.
Thank you. Another question for Dr. Wu is, what antibody was used to measure PD-L1 expression in the study?
Well, we didn't use the CPS, but we used another standard assay and different antibody and score system. And I think the assay is comparable to the standard assay comparing to the CPS. And we also show the CPS results in our study. So I think this is a relatively accuracy comparing to the standard detection.
Thank you. Actually, a question for either Dr. Wang or Dr. Wu is, do you guys have any safety data about using either ivonescimab or BNT327 in a preoperative setting to understand safety and exposure prior to surgery?
Well, in our cohorts for this 42 patients, 42 patients, and the 50% are de novo patients. And also there are another 50% of those patients are recurrent patients. And none of these patients received surgery afterwards.
Are there any plans with either of these agents to look at preoperative studies?
Preoperative studies, not yet, I think.
Not yet. Thank you.
Okay.
Any further questions from the audience for our authors?
Can I say something about the combination of those two target agents? when we designed these bispecific compounds targeting the VEGF and the PD-L1, especially the BNT327, and we hypothesized that when you block these two targets in the tumor cells, that will have some synergistic effect to treat those patients. And the most important one, I think, is anti-VEGF and can neutralize the angiogenesis, and also you can have more effective T cells and go into the tumor and also decrease the level of low oxygen environment in these patients. And when you use the anti-PD-L1 target therapy, and you can kill the tumor cells also the tumor cells, the secretion of the VEGF in these cells could be scavenged by this treatment. So this is kind of a combination of these effects. So I think we need more translational researches afterwards to demonstrate this mechanism.
Do you have tissue collected from this trial to be able to go back and retrospectively look at biomarkers? And how about serum? I'm just wondering about circulating VEGF levels and other biomarkers that have been studied.
So yeah, unfortunately, this is not a pre-planned collection of these samples in this cohort, but in the future, the phase 3 study already began, and we will do that afterwards.
Thank you very much. Dr. Tarantino?
Dr. Tarantino, Dana-Farber. I have a question for Dr. Liu regarding the SHR neoadjuvant trial. Were CT scans of the chest performed to monitor for ILD during the trial?
The ILD is interstitial lung disease. We can see that we just finished the first stage of this clinical trial, and 35 patients have finished the surgery. We have so far we haven't observed any grade of the ILD. As we know, in the phase 1 clinical trial, the ILD incidence is very low. About more than 300 patients, the ILD incidence is only 2.6%. So I think this is the most important advantage of this agent. Thank you.
Thank you. If I can just add, was the monitoring just clinical, or did you also conduct CT scans of the chest during treatment to exclude radiological signs of ILD?
So the monitor?
The monitoring
of the ILD. Yeah, in our study, we just conduct chest CT scan at baseline and before the surgery. During the treatment, we didn't perform the CT scan, but we monitored the symptoms related to the ILD.
Thank you very much.
Thank you. Any additional questions from the audience?
I have a question for Dr. Wu. Since the bispecific data looks really good for phase 1/2, do you have the plan for future studies like phase 3 and others?
Yes, in China, we already initiated a phase 3 study comparing the BNT327 in combination with nab-paclitaxel versus paclitaxel single agent in this first-line metastatic triple-negative breast cancer patients. The total sample size would be 360. So in the global, the phase 2 study is already initiated, and with different dose of the BNT327, and the phase 3 global trial would be initiated after the fixed dose would be designed, and the initiated global phase 3 study would be initiated in 2025, next year.
Thank you. question from the mic.
Thank you. I'm a breast surgical oncologist from Beijing, and I have a question for all presenters. Do you have any information about lifestyle, like physical activity, diet? I guess that will be very important for any outcomes you are looking at. Thank you.
Sorry, this was a question about diet and lifestyle information regarding which presentation?
I guess that could be applied to all presentations.
I don't know if anyone has collected data on diet and lifestyle from any of these trials.
Well, I think in the phase 3 studies, some secondary endpoints would be including the patient quality of life and also some like PRO. But I don't know if this is can answer your questions just focusing on the diet or lifestyle.
Thank you. I guess it would be interesting to provide some information like to what degree the patients might have for physical activity. Like even for some patients, they might have very low physical activity. I guess it's important for all clinicians to like get a sense about the lifestyle quality of the patients. Thank you.
Thank you very much. If there are no further questions, we will conclude our session this morning, and thank you very much to all the presenters and our discussants.